Okay, this video is what is the difference between the Warburg effect or Warburg effect and the reverse Warburg effect. Okay, and here it is right here. This paper, by the way, comes from Michael Lisanti, PhD. The paper is Reverse Warburg Effect in high-risk breast cancer patients from Cell Cycle 2012. So Lisanti is probably the best guy to read and study when you want to start learning about the reverse Warburg effect. He described it in 2009. He's got a very nice lecture on YouTube. If you go to YouTube and you type in Michael Lisanti reverse Warburg effect, he calls it metabolic coupling or parasitic cancer parasitic response with tumor stroma. So anyways, this is, I think, very interesting. There's going to be an AO coming out of this, academic orgasm coming out of this in just a little bit here. So to start out, the Warburg effect comes from Otto Warburg, the greatest biochemist of the 1900s, won the Nobel Prize in 1931. Supposedly, he should have won 3 Nobel Prizes. And he described that when you take a tissue culture of human cells and you deprive them of oxygen, they will have a tendency to develop cancer. You know, many of them will die from the hypoxia. Hypoxia is lack of oxygen, but some will transform into cancer, and that is called the Warburg effect. And they will thus have damage. The hypoxia damages the mitochondria, and subsequent to that, the cancer cell generates energy primarily then from anaerobic glycolysis. So here's the classical conventional Warburg effect. The cancer cell producing pyruvate is the end product of glycolysis, metabolism of glucose, and that lactate would then be expelled into the extracellular matrix, so pumped out of the tumor cell, and then it'll cause an acidic tumor microenvironment that suppresses the immune system around there, and that helps protect the cancer from the immune cells. Okay, that's classic Warburg effect. The W's are pronounced but like V's in a lot of European languages, and I'm sort of in the habit of pronouncing it like a V, so you can pronounce it as Warburg or Warburg effect, either way. All right, so now what is the reverse Warburg effect? Well, the reverse Warburg effect is that it comes out of a couple issues. First of all, some of the original cancer research was done on a two-dimensional flat culture of cancer cells. And there were also primarily cancer cells in some of these cultures and a lack of stromal cells. And when you have a two-dimensional culture, you'll have a tendency to make everything equal in terms of its exposure to oxygen and some of the nutrients and whatnot, versus in a three-dimensional tumor and an in vivo tumor. In vivo means in life, in a real animal, versus in vitro means in glass, in like a research setting. You'll have a three-dimensional tumor, and there'll be parts of it that are more or less hypoxic than other parts with more or less nutrient exposure or access than other parts. And so as more and more research was done on more three-dimensional cancer environments and in vivo cancer environments, they discovered this reverse Warburg effect, whereby the cancer cell, so here is the cancer cell, epithelial cancer cell, and it's basically transformed the stromal cell. Typically that's going to be a fibroblast. You'll usually hear this described as a CAF, cancer-associated fibroblast. And what's happened is, well, I'll show you better slides in a moment here, but just to get the general idea, the cancer cell releases hydrogen peroxide, H2O2, into the extracellular matrix, and this has an effect on the adjacent cancer-associated fibroblast, CAF cell. They'll often call them CAF, CAFs, CAFs. And that H2O2 causes oxidative stress. Oxidative stress just means the oxidizing chemicals are higher in amount than the antioxidizing chemicals. All right? So when the oxidizing chemicals are higher, they can damage tissue by oxidizing it. Hydrogen peroxide is unique in that it can travel some distance before it reacts with something, unlike some of the other free radicals, like hydroxyl radical, for example. That'll react so fast it could never have a distant effect. Okay, so anyways, through the use of oxidative stress, and the oxidative stress by the hydrogen peroxide released by the cancer cell, in effect mimics hypoxia in its effect on the cancer-associated fibroblast, and it'll damage it. It'll damage its mitochondria. Its mitochondria will often be reabsorbed. When phagy means to eat. Mitochondria is abbreviated mito, so mitophagy or mitophagy means the mitochondria sort of eats itself. It's digested by the fibroblast, will digest its own mitochondria, and then they'll release chemicals from their autodigestion of their mitochondria and even other parts of the cell. Okay, now here's the AO, the academic orgasm of this talk. The tumor stroma cell, the cancer-associated fibroblast, the CAF, it will not only release lactate, as you would expect from anaerobic glycolysis, it'll also release ketone bodies like 3-hydroxybutyrate. And that's what's funny. Why is that funny? Why is that interesting? Why is that a huge statement? Because there's a lot of so-called experts on the internet going around saying ketone bodies are a great thing to feed cancer patients. And I can tell you this, Lisanti would 100% disagree with that, and I think it's a bad idea too for additional other reasons. So what he's saying is ketone bodies are one of the cancer's favorite fuels, and this reverse Warburg effect that's now been studied pretty extensively since 2009. Lisanti, who's like the world-class expert on the subject, says no, ketone bodies are a bad idea. They help cancer to grow more faster, more extensively. Bad idea. So just so you know, this is, and he'll say it if you watch his lecture on the internet, Lisanti's lecture about metabolic coupling with cancer and reverse Warburg effect. He'll talk about it. Here it is. The ketone bodies accelerate cancer growth. All right, trust me, that's a huge, giant, big statement in the world of cancer biology and cancer nutrition and cancer pathophysiology. Okay, what else are little things to notice about it? So that was the big E enlightenment thing in this whole talk. Little E enlightenment things. Okay. MCTs are monocarboxylate transporters, so transporter things with one carboxyl group on them. And so the 1 is on the cancer cell, and the 4 is on the monocarboxylate transporter number 4, okay, for releasing lactate and ketone bodies outside of the cell, okay? And MCT1 is for receiving them, taking them in to the cancer cell. That's going to come up again. Okay, here's the same thing as on that previous slide, but a little bit fancier. So CAF means cancer-associated fibroblast. Here's the breast cancer cell. TAM is tumor-associated macrophage, but we're not going to talk about those. Those are not as important. They don't really matter for our talk here today. And our talk here today, what we're going to talk about is this cancer cell releases reactive oxygen species in the form of hydrogen peroxides that can diffuse out of the mitochondria, can exit the plasma membrane of the cancer cell. This is the cancer cell, can pass across a small distance between the cancer cell and the cancer-associated fibroblasts. They will then exert an oxidative effect of reactive oxygen species, and this oxidative stress is like a hypoxia effect upon the fibroblast, and that will induce increased HIF to be released. HIF is hypoxia inducible factor, okay? The 1-alpha part really doesn't matter for our purposes. HIF, hypoxia inducible factor, and that activates a whole cascade of processes. It's a transcription factor and causes the cancer fibroblast to run a whole bunch of new enzymes to produce them from its DNA. So there's an epigenetic effect, a transcription effect. In addition, it'll increase something called nuclear factor kappa B, and that will have additional effects upon this fibroblast. It'll start to digest itself, especially its mitochondria, so it develops mitochondrial dysfunction. It's now like a Warburg effect. And it will then metabolize glucose to pyruvate and then expel it out of the cell and decarboxyl and then change it into lactate, and that's taken up by the cancer cell and can, metabolize it. It can run pathways on it. It can send it into Krebs cycle. Okay, so what's happening now, in a sense, like what is the big picture? What does this all mean? It means that the cancer cell has turned the cancer-associated fibroblast into its slave. Okay, that's why it's called parasitic coupling. The metabolic coupling is that the cancer cell is working with the fibroblast to make the cancer worse. And it's parasitic because it's not a fair deal. The fibroblast is being destroyed. Its mitochondria is digesting itself, and it's also going to digest other parts of itself. And the cancer cell is stealing all of its nutrients. So basically, the cancer cell is ripping off the fibroblast and taking advantage of it. And it does this with almost like the way it's able, how is it able to control the fibroblast and force it to accept its will, is by releasing H2O2, hydrogen peroxide, onto it. And that hydrogen peroxide is damaging the fibroblast and turning it into a slave for the breast cancer cell, okay? Some of the things that happen as it undergoes autophagy, it will also diminish the amount of this protein it has called caveolin. And that's relevant because Lisanti's lab can test for caveolin, and if caveolin is diminished, it means this patient has a much worse risk of a poor outcome, of earlier death. And so that is a test that Lisanti feels should be used more often in evaluating cancer patients. Okay. So this is the reverse Warburg effect. It's called the reverse Warburg effect in the sense that typical Warburg effect meant the cancer cell was undergoing anaerobic metabolism and expelling lactate from itself. The reverse Warburg effect means that it's actually the stromal cells, the cells around the cancer, that are undergoing anaerobic glycolysis and releasing lactate outside of themselves, okay? And there's other nutrients coming from this fibroblast that the cancer cell is stealing all of them. So basically, it's just taking all the nutrients and the energetic efforts of the fibroblast and to use it for its own benefit, the cancer cell. And so what I'm trying to say is cancer is not a single mutation. The somatic mutation theory doesn't make sense, whereas the metabolic theory makes total sense. And this is not just one little pathway. It's a whole bunch of pathways, all activated, that are going to promote the growth of this cancer cell. And in addition to lactate being passed between the cells, it's also ketone bodies. And so that's a rather extraordinary statement. Okay, our results indicate, here it is, Lisanti talking again. Here it is. Our results indicate that ketone bodies can effectively serve as a source of energy for cancer cells. Ketone bodies can serve as a substrate for neuronal oxidative metabolism even during hypoxia, because oxidation of ketone bodies by neurons is not impaired during hypoxia. Ketone bodies require far less oxygen for their use in mitochondrial oxidative metabolism, and they thus behave as a superfuel. Okay. In addition, we have shown that administration of 3-hydroxybutyrate, a ketone body, increases tumor growth in the human breast cancer. Okay, so what this is basically saying is ketone bodies, ketogenic diet are a very bad idea for cancer patients. This is all coming from the work of Lisanti. We know that animal protein is a disaster for cancer patients from the work of T. Colin Campbell, because it activates all the growth factors, and that's what I'm going to show you here too. So this is MTOR. MTOR, to pronounce it out, is mammalian target of rapamycin. And it's a fancy way of saying the name of this nutrient sensing. It's also sometimes called mechanistic target of rapamycin. Rapamycin is like an antibiotic that blocks it. But the point I'm saying here is mTOR is like a building contractor. It's a nutrient sensing pathway that's like a building contractor, and it's waiting to have enough proteins available, because you need proteins to build cells and cellular components. And it's the rate limiting step is leucine, and that's the most important one, but also the other branch chain amino acids like isoleucine and valine, in particular leucine though. Methionine is also an essential amino acid. So until it gets that leucine and the other amino acids, mTOR will not send the signal to build. But once mTOR has got those amino acids, it can tell the cell to grow and to replicate. So you don't want to turn on mTOR. Okay, and mTOR also will activate the hypoxia-inducible factor-like pathway, in the sense that that pathway can also be used for rapid growth. When a cell wants to grow rapidly, it focuses on synthesis, biosynthesis. You got to double yourself if you're going to divide in half. So you got to make tons of DNA, tons of plasma membranes, tons of the nucleic acid. Every cell, human cell, has 3.3 billion DNA base pairs, which means 6.6 billion nucleotides have to be synthesized to replicate the cell's DNA material. All right, that's a ton of material. Okay, so you run all these biosynthetic pathways, which actually can run on anaerobic glycolysis, because a lot of molecules are being shunted off to the pentose phosphate pathway, for example. Krebs cycle can run partially in reverse, for example. So what am I saying here is, this is exactly what T. Colin Campbell say. What I'm basically saying here is a confirmation of the work of T. Colin Campbell, that it seems to be that he's right. If you're going to stop cancer with anything, probably one of the best things you could do was stop activating mTOR. And what activates mTOR? Primarily animal protein with all that leucine. Animal protein has a lot more leucine. Its amino acid composition is different than plant protein. And other things that get activated by activation of mTOR. It'll also increase those glucose type 1 transporters to go to the plasma membrane to suck up more glucose type 1 transporters. Those are the ones, GLUT1s are not insulin dependent. So they're like a vacuum, sucking up all the glucose they can get out of the passing blood. In addition, you're going to ramp up HK2. HK2 is hexokinase 2, which phosphorylates glucose when it first comes into the cell, makes it glucose 6-phosphate, putting that big negative charge of the phosphate on there, which traps the glucose into the cell so it can't get out. But the point being is hexokinase 2 is very unique. We've talked about it before. I'll show you a picture of it later. And it'll ramp up other glycolysis enzymes like PFK, phosphofructokinase. So what I'm saying is, this is not one mutation. Somatic mutation theory is bogus. This is metabolic theory of cancer, and it makes a lot of sense. You're basically activating this metabolic growth pathway. Cancer is all about growing. And again, this is as if the cancer cell, a eukaryotic specialized cell, is transformed into behaving like a primitive bacteria that only cares about growth, and it's selfish. So we talked before about what's the difference between a prokaryote and a eukaryote. Pro means before, karyote means nucleus. So this is like a bacteria. It's pro means pre, so pre-nucleus cell. And a eukaryote, eu means true in Greek, so this is true nut. Karyote means nut, and we refer it then to meaning nucleus. So the true nut is the cell with a nucleus. But we're going to take it a little further. The real unique thing about a eukaryote is not the nucleus. The real unique thing about a eukaryote is the mitochondria. It's having the mitochondria that makes eukaryotic cells so special, because mitochondria can produce 18 times as much ATP as glycolysis alone can produce under anaerobic conditions. So any cell that's going to become multicellular is going to have a lot more tasks to do to run a multicellular organism, so it needs mitochondria for energy. Once a cell loses its mitochondria, it becomes, in a sense, sort of primitive, like a bacteria like a prokaryote. Okay, here's just another reminder of the basic biology here. Anaerobic bacteria are prokaryotes. They'll run, let's say, glucose to pyruvate, expel lactate. A normal human cell runs glucose to pyruvate through glycolysis, then it sends it into the mitochondria to run Krebs cycle, and then through electron transport along the intermitochondrial membrane with oxidative phosphorylation, it makes tons of ATP. It expels CO2, carbon dioxide, as a waste product, and that's normal human oxidative metabolism. Now, a cancer cell, classically with the Warburg effect, is a damaged cell whereby its mitochondria is injured and it can't run oxidative phosphorylation, to make ATP. It may run Krebs cycle in reverse. I got reverse Krebs here. And we all know glucose can partially feed the energy needs of a cancer cell, also feed some of the biosynthesis needs. But the big problem with the cancer cell is it's also fed from below, sort of the bottom end of Krebs cycle, if you will, by the very common abundant amino acid glutamine. And glutamine gets converted to glutamate, and glutamate to alpha-ketoglutarate. That's also why a lot of people think there's a problem. I'll talk about it some other future time with the glutamate. That's a separate issue, but just remember that it's not good. Okay, here is the appearance of hexokinase 2. So hexokinase 2 takes the place of hexokinase 1 in the beginning of the glycolysis. And hexokinase 2 is unique in that it attaches to the outer mitochondrial membrane. And it also attaches to something called VDAC, voltage-dependent anion channel. And what it does by attaching to VDAC, it locks that outer mitochondrial membrane into a survival mode where it will not undergo apoptosis. So this helps confer immortality upon the cancer cell. And that guy, Peterson, he's the one who really figured out a lot of this stuff about how hexokinase 2 is the most important transformation of an enzyme step in all of cancer. So actually, if you care about cancer, you want to know that. Hexokinase 2 attaching to the outer mitochondrial membrane and VDAC, and it gets whatever ATP is coming off this mitochondria, it gets first dibs on it. This is from this paper, Emerging Glycolysis Targeting and Drug Discovery from Chinese Medicine and Cancer Therapy by Jinggang Shen. Okay, so now we just talked about this, that you can also feed cancer with glutamine from below. That's why I don't think you can manage cancer that effectively by just trying to deprive it of glucose or just trying to deprive it of protein in general. And some protein can be taken from the body and sent as glutamine to the cancer, and that's part of what cachexia is all about. The person loses weight. That's part of what that's all about. There's more to it than that, but that's part of it. Just simply know that total protein by itself isn't going to win the game. Okay, here is bacterial life cycle. Like we said, the cancer is transformed from a eukaryotic cell, part of a multicellular organism. And to be part of a multicellular organism, a cancer cell has to do tons of work. If you're going to be a liver cell, you got to make bile. You got to detoxify things. You got to sense blood glucose, maintain blood glucose, maintain glycogen stores. There's a lot of work to do. A cancer cell is like an anaerobic bacteria. It just says, screw this. I don't want to be part of the liver. I don't want to be part of any organ system. I'm just going to try to survive. I'm going to go get myself a new apartment somewhere else. So the cancer cell, it can lay dormant until it gets its food, and then all of a sudden grow rapidly. A cancer can lay dormant for years and then grow. That's why once you've had diagnosed cancer, you want to maintain this optimal low-fat, low-sodium vegan diet the rest of your life, so you never want that cancer to come back. And the people who have maintained good health habits, some of them, are alive 40-something years later after metastatic cancer, like Ruth Hydrich with metastatic breast cancer. Okay, anyways, well, once the cancer gets a food source, it'll rapidly multiply like crazy. It'll often deplete its resources. This is, in particular, its life cycle on a culture plate in, microbiology lab. And then after that, because of its depleted resources, it'll progressively die off. But that's kind of how a cancer goes. It's a very stupid strategy in a sense, because it kills the host. The person dies from the cancer growing too rapidly. Okay, you also want to minimize insulin. So by avoiding high fat, you minimize insulin resistance. Also by minimizing stress. Because at hyperinsulinemia, the insulin's a mitogen, meaning mitogenic, meaning that it stimulates a cell to undergo mitosis, to divide. Okay, we talked about the tumor cell secreting H2O2, hydrogen peroxide, into the extracellular matrix. We talked about the H2O2 causing oxidative stress, which is perceived by the fibroblasts, the cancer-associated fibroblasts, as hypoxia. So it's called a pseudohypoxia effect. And then it releases HIF and NF-κB. So HIF is hypoxia-inducible factor. NF-κB is nuclear factor kappa B. All right, and then these initiate all those things that happen subsequently in the cancer-associated fibroblast. So Dr. Lisanti in 2009 coined the term reverse Warburg effect, and it's also called parasitic metabolic coupling. And Lisanti has a good YouTube video if you want to see him talking himself. It's available on YouTube. It's good. And he basically says, the cancer is being a parasite, and the tumor stroma, the fibroblasts in particular, are making lactating ketone bodies as well as glutamine to feed the cancer cell. Okay. And also, you can get a similar lactate shuttle effect in skeletal muscles. The fast-twitch glycolytic cells will send lactate out to the slow-twitch fibers, which then function aerobically. And in the brain, in a sense, like the fast-twitch, well, the way it works in the brain is the astrocyte is like the helper slave to the neuron. Okay, but the relevance, why did I even mention all that? This idea that these are established metabolic strategies that are used by other parts of the body. This is not just some weird mutation thing that's happening in a cancer cell. It's basically the cancer cell is taking over stuff to make itself able to grow faster. All right, so we know that, high-fat diet causes red blood cells to stick together, rouleau formation. When they stick together, they're slower to pass through the capillary. Blood pressure has to go up, leads to atherosclerosis over time. The RBCs stuck together are less able to deliver oxygen, so that's going to cause tissue hypoxia. Okay, when you feed people high-fat diets, fat in the blood goes up, and they get less oxygen to their tissue. Peak lipemia with sat fat is the black line here, worse from about 4 to 7 hours. With omega-6 vegetable oils, it lasts even longer. When there's no fat in the diet, they don't get any of that stuff. And the point is, all of that's causing tissue hypoxia. So you would expect those things to worsen the risk of cancer. We talked about how when there's inhaled foreign body like asbestos, you get an inflammatory fibrotic reaction around it, formation of scar tissue, and that drops oxygen delivery to the tissue cells trapped within. The hypoxia might induce cancer in them. That's how asbestos can induce cancer. We talked about how in the liver, just from an infection, hepatitis B, hepatitis C, non-alcohol fatty liver, non-alcohol steatohepatitis can cause progressive inflammation. Fatty liver can lead to this inflammation, as can those infections, as can alcohol. And as you get the scar tissue, secondary to the inflammation always leads to scar tissue. And the scar tissue, like let's say collagen fibers all placed around, they entrap some normal cells, and those cells can sometimes be transformed into cancer. So what I'm doing is I'm giving you a bunch of different examples of how tissue hypoxia, tissue ischemia is lack of oxygen, can occur through these converging pathways and lead to the generation of cancer. And that's why earlier today, if you saw my earlier lecture about is Dr. Esselstyn crazy, what I'm saying is maybe if he's right about being able to hyperoxygenate tissues by eating leafy greens all day, then that might be a way to maintain oxygen delivery to these tissues and decrease the risk of cancer, which is an extraordinary useful thing if it's actually true. I haven't yet seen the paper to confirm what he said, but like I said, I've never seen Esselstyn say something that wasn't true, so it probably is true. I just haven't found the paper yet. And I did watch a couple of his lectures, and I didn't see him mention it. I went to Esselstyn's website, and I didn't see a reference for the paper there, so... and I heard that he kind of got some of these ideas from Nathan Bryan, so maybe I'll find the paper as I study Nathan Bryan some more. Nathan Bryan, PhD, is sort of like the world expert on nitric oxide, so I'm going to bet you the information's out there somewhere, but I haven't seen it yet, so I'm still a little bit of a doubting Thomas until I see that, okay? We talked before about a normal capillary basement membrane. Here's the red blood cells going through a capillary. The blue circles are the oxygen coming off of the red blood cells, off the hemoglobin. They diffuse over to the tissue. But when you have hypertension or diabetes, you get capillary basement membrane thickening. So this is a capillary. These are the endothelial cells. They're sort of spindle shaped. That's their nucleus. These are the red blood cells going through the capillary. This is the capillary basement membrane. The green stuff is this vascular smooth muscle cells around the capillary. And what I'm trying to say here is that when you have capillary basement membrane thickening due to chronic diabetes, chronic hypertension, less oxygen gets to the tissue. So that's why you want to get your diet together, the sooner the better, so you minimize your capillary basement membrane thickening. That's why you also want to minimize your dietary fat intake, so you're going to maintain optimal oxygen delivery to your tissues. And why do you want optimal delivery to your tissues? So you don't induce a Warburg effect, so you don't induce cancer-causing effect, and an effect that also helps cancer to proliferate. Okay, this was just a quick review of what a cancer cell does. Normal cell, it mostly wants to make ATP because it's got a ton of work to do. A liver cell wakes up in the morning and it's got lots of work to do. It's been working all night. A cancer cell says, screw this, I'm not doing any work. I'm not part of an organ. I'm just living for myself. It's a selfish cell, like an anaerobic bacteria. It wants to just do tons of synthesis because it has to make a copy of everything inside of it so it can divide. Before it divides, it has to double itself, and that means tons of synthesis. It'll make some ATP. Even better, crack the whip and turn the adjacent fibroblasts into slaves and just take the macromolecules they've already made and use that for its own biosynthesis equivalent so it can divide. Cells that undergo the reverse Warburg effect have a worse prognosis. And this is an important mechanism seen in breast cancer cells. So here's Lisanti's paper again. I'm just showing you the same thing again. So seeing it again, it'll make more sense to you now. So that here is the reverse Warburg effect, where here's the epithelial cancer cell, the ductal breast cell that's cancerous, and it will be getting lactate and ketone bodies from the tumor stromal cell, the cancer-associated fibroblast. And this is the reverse Warburg cell. And notice I wrote worse prognosis there, under traditional conditions, of course. All right, and so the conventional Warburg is just the cancer cell is the one undergoing the anaerobic glycolysis and expelling lactate. So very interesting, I think. And this was just, again, the same diagram, a little more sophisticated with more detail about here's the breast cancer cell, here's the transformed cancer-associated fibroblast, CAF, and the hydrogen peroxide being released from the cancer cell, inducing oxidative stress upon the fibroblast. This is sensed as a hypoxia-like equivalent, so it releases hypoxia-inducible factor as well as nuclear factor kappa B, and that's setting off a cascade of protein synthesis and subsequent reactions to cause autophagy or mitophagy. The mitochondria is digested by the cell, and then you have dysfunctional mitochondria. So you undergo anaerobic metabolism of the glucose, convert it to pyruvate, expel it as lactate. The lactate's taken up by the cancer cell, and it is able to metabolize it because this particular cancer cell does have some oxidative metabolic capacity. So anyways, this is the reverse Warburg effect, and also the ketone bodies are also sent from the fibroblast to the cancer cell, and thus the ketone bodies are a fuel for the cancer. So anyways, that's what the reverse Warburg effect is all about. I thought that was interesting. Hope that was interesting to you.